Hello everyone, my name is Mahmoud Teran. I am an undergraduate at Wayne State University, and the title of the research project I'm involved in is Utilization of in vivo longitudinal PET imaging to assess Class II HDAC activity following long-term extinction fear memory in female and male rodents. As you may already know, the development of memories, including those throughout extinction learning, require neuroplasticity for encoding and retrieval to occur. Interestingly, environmental factors can have a profound effect on gene expression, as epigenetic changes can play a critical role in the development of memory formation and updating. Additionally, epigenetic changes related to learning ultimately manifest in behavioral changes reflective of the learning. As I stated earlier, environmental factors can affect gene expression, ultimately repressing or expressing genes. This is regulated by HDACs and HATs, as they are responsible for expressing or repressing genes. HDACs, which stand for histone deacetylases, work by removing an acetyl group from histone lysine residues, which results in a closed chromatin conformation where RNA polymerase II is unable to bind and genes are repressed. HATs work by doing the opposite, and this figure provides a visual representation of that. Having said this, changes in activity levels of HDACs is indicative of learning, specifically related to acquired long-term fear memories. The purpose of the study is to assess changes in Class II HDACs before and after extinction learning via PET imaging to understand the neurobiological mechanisms of extinction learning of acquired long-term fear memories in female and male rodents. The subjects used in the study were wild-type Worcester rats. 18FT Faha was used as a radio tracer. PET and CT imaging were conducted, and Rat W Schiffer Atlas was used for co-registration. The experimental design was as shown. 18FT Faha was used as a HDAC PET tracer, as it is a substrate for Class II HDACs. Therefore, it is reflective of Class II HDAC expression changes before and after long-term fear memory. Our preliminary results show that rats subjected to shock show increased freezing behavior during acquisition, and this is clearly shown in the graph on the left, while the graph on the right reflects changes in extinction learning. PET imaging allows for visualization of changes in brain activity. These changes can be quantified by looking at the standard uptake values of 18FT Faha in specific brain regions. Our preliminary results also show decreasing expression activity levels from pre to post PET scans for males, while no decrease for shock females, which indicates that different mechanisms between sexes regulate fear extinction. The behavioral results indicate female and male shock subjects acquired and extinguished fear behavior, but no sex differences. Additionally, preliminary analysis of PET imaging results indicate a significant interaction effect between pre to post Class II HDAC expression activity, sex, and shock conditions. With decreasing expression activity levels from pre to post PET scans for males and no decrease for shock females, this could indicate that different mechanisms between sexes regulate fear extinction. As part of my project, I have been conducting additional PET image analysis to show the extent of HDAC activity in both sexes following long-term fear.